So let's take a look at mitosis. One of the things we talked about, we said that mitosis is going to be the division of the nucleus. But let's look at what's going on here within this cell. So the first thing that we'll see during interphase, we have DNA in the form of chromatin, that loosely coiled form. Nuclear membrane is still intact. You can see that the nucleoli is still intact, and then you can also see the plasma membrane here on the outside. Is this structure here, which is known as the centrosome. So the centrosome is composed of these pair of centrioles. So these are centrioles. Centrioles have what's known as these different microtubules. So these microtubules, they are going to play a key role in moving these different sister chromatids apart. So these sister chromatids, they are going to be held together by what's known as a centromere. So the centromere, that's this part here that's keeping these two sister chromatids held in place here. So what will happen, so this is during early prophase, and this is during late prophase. So if you look at some of the key differences between early versus late, look at the formation of the nuclear envelope here. The nuclear envelope is going to be gone during the late phase. The kinetochore microtubules, that's what's coming off of here, and that's what's attaching to those centromere to start to pull these different sister chromatids apart. So metaphase is that part in which all these different sister chromatids are aligning here down the middle. They form that what's known as the metaphase plate. So that's what they're distinguishing here. That's the metaphase plate. Anaphase. So anaphase is whenever we start to pull those sister chromatids apart. And the terminology that they use to describe these are now what's known as these are the daughter chromosomes. So telophase is going to be here, starting to get the formation of that cleavage furrow. That cleavage furrow by what's known as actin. So remember, cytokinesis, it starts during late anaphase, and then it is going to end after telophase. Cytokinesis, remember, that's the division of the plasma membrane. And so you can see you're starting to get the formation of the nuclei are starting to form again here. And you can also see the formation of the nuclear membrane is starting to form again here. I'll be very brief about this for the control of cell division. This is what we talked about, those different checkpoints. We talked about the G1, and then we talked about the G2/M checkpoints. These are like regulatory proteins here, cyclins. These are regulatory proteins that are going to be involved in moving from one phase to the next. Those are the only key things here I want you to be aware of for the control of cell division. 3.11, messenger RNA carries instructions for DNA for building proteins. So we look at, this is what's known as the central dogma of biology. During transcription, we get the formation of our mRNA. And so this is specifically what's known as pre-mRNA because it's not in its mature form. That mature form is going to have the 5' cap as well as that poly A tail, and another feature is that we're going to splice out what's known as our introns. So introns get spliced out, and then exons going to be expressed. So that is what is going to be left when we have our mature messenger RNA. And that is what's going to exit out through that nuclear pore, and then over here is where we get translation occurs, where we are converting that messenger RNA. We use the coding that's in that strand in order to create our protein here. So keep in mind here that the location of where these things are occurring. Transcription occurs in the nucleus, and translation is going to occur in the cytoplasm, whether that is on the rough ER, the ribosomes in the rough ER, or whether that is on a free-floating ribosome. And so depending on where that protein goes, if it needs to be located or if it needs to be moved to, for instance, like the plasma membrane, we're going to use the rough ER in order to transport it. However, if it's like a cytoplasmic, some type of protein or some type of enzyme, we'll just synthesize that within the cytoplasm. The different types of mRNA. So one that we talked about was messenger RNA, and then another one is going to be our ribosomal RNA, so that is going to be part of the ribosome. And then the last one is the tRNA. So tRNA is going to have a role during translation, and we'll look at that here a little bit later. But first, let's look at some of the details here that's occurring during transcription. We talked about DNA polymerase during DNA replication. However, this is what's known as RNA polymerase. So RNA polymerase is going to bind at this particular region over here, which is known as the promoter region. It recognizes this promoter region, and then from there we can start to create our messenger RNA. There's 3 phases for transcription, and you can see them here: initiation, elongation, and termination. So this first step over here, this is initiation, right? Binding to that promoter, and it's first starting this process here. But once we get to elongation, we are now creating our messenger RNA that does not have thymine. It has what nucleotide? It has uracil instead. So we still have that complementary base pairing, but instead of using thymine, we use uracil. So it elongates this certain transcript here, and then the last phase is termination. So during termination, you have the termination signal, which is going to be found here at the very end. We have that termination signal, so we'll make our transcript, and then that RNA polymerase is going to hop off. And this here is just showing the particular complex, which is known as the DNA RNA hybrid region, because we have not released that mRNA yet. And so one of the things we talked about was the processing of mRNA. We use it's pre-mRNA before we use our spliceosomes in order to remove the introns, and then we only have our exons expressed, and then other key parts for the mature mRNA was our 5' cap. So our 5' cap, and then our 3' poly A tail. One other thing to make note of is what's known as the transcription factors. They are going to bind to a particular region where a segment of DNA that needs to be read to form our transcript, they are going to bind to where the histones are. So they'll bind to that histone in order to unravel that particular region, and from there, then you can get the movement of RNA polymerase in order to start to make the messenger RNA here.